All right, everyone. Good evening. The last one was a false alarm. This one is for real. All right. Good evening and welcome to the 14th Annual Image Awards. If you've ever been to an Image Awards, you may realize that you may have not seen me before. This is because, hello, I'm new. I'm Christine Pace. I'm the new Education Outreach Coordinator here at the KI. And it is my pleasure to host and be your MC for tonight. So, fun fact, there is, we wanted to make sure we shout out for our judges. We had 159 submissions, and out of those, the 10 that got selected are displayed before you tonight. And we are very grateful to all our judges who made that possible, some of them being here tonight. Another thing, the presenters may know, but the audience may not. We have a little thing called the People's Choice Awards. This QR code, if you would like to take a picture of it now, there'll be another one at the end of all the presentations. So feel free to, if you don't do it now, you'll get it later. You get to choose your 2 favorites. And the reason why this is important is this event is sponsored by Fujifilm. And they were so generous that they gave us 2 camera sets. So the top 2 will get a camera set to take home. But you are already winners just by being here tonight. So it's always nice to get a little extra something. All right, shout out to our sponsor, Fujifilm's Visual Sonics. Some of you may know this, but I previously was a middle school science teacher. And I'm really surprised the similarities between middle school and MIT University life. First of all, we have to have our morning announcements, now more like afternoon announcements. But if you know a grad student who is pursuing life sciences or biomedical engineering degree outside the US, and they have a passion for discovery of science, please, please, please encourage them to apply to the Koch Institute 2004 Angelica I Am a Young Scientist Award. If you know someone, make sure to take a picture and send them the QR code. The deadline is Friday, May 31st. So it is a fantastic ceremony if you have not had a chance to visit. The other event that we have is our next symposium on June 21st for tumor heterogeneity and drug resistance. It's a 22 annual Koch Institute symposium. So if you love learning about science and are here, please make sure you sign up for the Koch symposium, June 21st. Any more information? Now everywhere has social media. We are on Facebook, X, formerly known as Twitter, Instagram at the kocinstitute.mit. So any pictures that you take, if you want to post us or tag us in, we are happy to present you. And feel free to explore more on our website at ki.mit.edu. Okay, now the morning announcements are over. Let's get to the celebration. Ooh, wait, one too fast. So some of you may know the Koch Institute is celebrating its 50th anniversary. We have to acknowledge our past and especially the role that the Image Awards have played into the culture that we have today as MIT. Dr. Alex Florentino started this event as a way to recognize and publicly display the extraordinary visuals produced by life scientists and biomedical research at MIT. My predecessor, Erica Reinfeldt, built on the foundation and lovingly curated this popular and prestigious annual event as we know it today, and are here to celebrate. It is my honor to continue that legacy that Erica Reinfeldt put so thoughtfully cultivated and punnily executed time and again. I will not have as many puns. I am sorry for that in advance. The bridge between science and art is an interesting one. Science is humanity's attempt to make sense out of the universe, while art is creativity expressed into reality. The great thing about the Image Awards is that they serve as a culmination of grand ideas visually manifested into action. It is the humanity behind our research that makes all these images that you're about to see tonight impactful. All our speakers are here to show their research in creating understanding for our internal world and imparting change for the better, adding to the legacy of those who have come before them. These images tell a story. And after all my beautiful words, I'm going to kind of take a sharp left. Due to my nerdiness, I can't help but see Dungeons and Dragons as the common thread in this year's winners. So let us start our adventure with our first presenters giving us our first dragon of the night. Without further ado, Brian Wong and William Jang.
Good afternoon. I'm Brian. I'm a senior in biological engineering in the Anderson and Langer labs. I'm here with my mentor, William, who is a PhD candidate. And together we'll be presenting on our Midnight Fibrotic Dragon. So one area that the Anderson lab is interested in is medical devices and implants. Medical implants exist on a large spectrum and have been used to treat a large range of diseases. So just a few examples up here are pacemakers, which are able to send electrical signals to the heart in order to treat heart arrhythmias, and drug depots that are able to release a scheduled dose of any drug without the need for repeated injections. Implants have also been used for structural and cosmetic reasons, for example, with breast implants. Once the implant is inside the body, you would hope that a helpful device would be accepted with open arms and welcomed by the immune system, but unfortunately, it is not. So when the body encounters an unfamiliar object, it begins something known as the foreign body response or fibrotic response. During this response, the immune system will recognize the implant as foreign, it'll attach to it, and it'll begin attacking the implant. It'll also recruit other cells to deposit extracellular matrix and collagen onto the implant, essentially walling it off from the rest of the body. This process, known as fibrosis, evolved as a response in order to protect the body from foreign objects to stop them from causing further harm or infection. However, as you can imagine, this is a problem for our medical implants. So using our earlier examples for pacemakers, this fibrous encapsulation can prevent pacemakers from sending their electrical signals. It can prevent these drug depots from releasing their drugs. And for soft devices, such as breast implants, it can actually cause complete rupturing of the implant. So therefore, we aim to prevent this fibrous encapsulation by creating a polymer coating that is able to tell the immune system that the implant is safe and prevent it from recognizing it as dangerous. And by doing so, we hope to improve the function and the longevity of medical implants.
So how are we actually doing this? Our approach is to take some immunosuppressive chemicals that we've discovered in the lab and actually transform these into a solid state coating that we can then apply onto the surface of different medical implants. And the effect of this is that we can actually generate essentially immunosuppression upon contact and effectively reduce the safety concerns that you have associated with systemic immunosuppression. And in order to test this, we generated a library of different types of polymer coatings and put them onto different implants. And then we implanted them into mice for at least a month before we explanted them and analyzed the tissue that ends up depositing on the surface. And long story short, you can take something like this and turn it something more into this. So as you can see with our lead candidates, we're actually able to dramatically reduce the amount of fibrotic tissue that forms up on the surface of the implant, allowing us to ensure a clear line of communication between the implant and the patient's body. And for some sort of technology transfer or a scientific publication, this before and after image that we're illustrating here is kind of what we really want to see. But what we found kind of interesting was actually this image that we captured and ended up being the image award contest winner. And what we see here is actually this dynamic fibrotic process in action. This partially fibrosed implant that we're able to capture shows these billowing cloud structures, which are actually swarms of immune cells that are sort of the first line of defense that are crawling along the surface of the implant and flanked by this dragon-like structure that's actually collagenous tissue and newly formed blood vessels that can bring further immune recruitment to the implant site. And so with that, we hope that you guys enjoyed a little snapshot of the work that we're doing upstairs. And we want to thank the people that made this possible, including our collaborators, our professors, Dr. Robert Langer and Dr. Anderson, and then of course our funding sources, and of course the Koch Institute for providing this great environment to do this work.
Thank you so much for being part of our work today. That's going to happen about 9 more times. So up next, well, thank you to Brian and William. Continue with our D&D theme. As we venture to our next image, we enter the dragon and their fiery response to hard to treat cancers. Up next is Padmini Pillai, and she is here to tell the tale of the fiery immunogenic death for cancer. Padmini.
Thanks, Christine. Hi, everyone. So before I tell you about cancer, I'm going to talk to you about death, but specifically cell death. So cell death is an essential process for our body to function normally, to maintain what we call homeostasis. So old cells or damaged cells die and are replaced by new ones as a form of self-renewal. And this kind of self-renewal process, which I'm sure you've thought about before, it's highly coordinated and regulated, and it's what we call programmed cell death or apoptosis. But it's also immunologically silent. So in a sense that when these cells die, you're not setting off any alarms, you're not triggering any sort of immune response. And so this kind of cell death is very important, and it's also important for growth and development. And hopefully this will bring back some memories of childhood. If you ever had any tadpoles, and if you watched them develop into frogs, you would see that their tails would start to shrink and disappear. And this process was also because of programmed cell death or apoptosis. So the cells in the tail would neatly package themselves up for removal, and they would get taken back into the body. And so those cells were dying by the silent process. It was just kind of a silent exit, right? Unfortunately, a lot of cancer treatments trigger this kind of silent death. And you can imagine why you don't want that to happen, because you want your immune system to recognize that cancer and be able to attack it. But unfortunately, a lot of cancer treatments trigger this kind of silent death. And when those cancer cells die this silent death, they can even send signals to neighboring tumor cells to tell them to divide and replace them. So not ideal. But we've also evolved a number of ways, much more flamboyant ways for cells to die. And you can imagine why. So for example, if a cell is exposed to a toxin or it's infected by bacteria or viruses, that cell can commit suicide to prevent the spread of that pathogen, but it also needs to alert the immune system that there's an infection and that there's something happening. And so this is why we have what's called immunogenic cell death. So during immunogenic cell death, the cell releases a firestorm of signals that activate the immune system. And then these activated immune cells can go back to the site of cell death and can trigger an immune response. So you can imagine this is the kind of response that we would want to treat cancer, for cancer to undergo this immunogenic cell death. But unfortunately, cancers have figured out ways to avoid this from happening. So cancers can inhibit immune activation, and they can also mutate to evade immunogenic cell death, ensuring that they can proliferate unchecked. So what do we do about this? Well, this is where mRNA technology can really help to treat cancer. So many of you may be familiar with mRNA technology, especially if you got one of the mRNA COVID vaccines, but it's a really elegant strategy where mRNA can be designed to to encode for whatever protein you want. And so we can design this mRNA, and then we put it inside a bubble called a nanoparticle, and then we can inject a bunch of those nanoparticles. And when the nanoparticle gets inside a cell, that mRNA is translated into protein using the host cell machinery. So in other words, the mRNA provides instructions to the cell, and then that cell will make whatever protein we choose. And in the Langer lab, we have technology where we can deliver mRNA directly into cancer cells or into a tumor. And so you can imagine now we can deliver mRNA that will express proteins to trigger this immunogenic cell death, to kill the cancer and also trigger an anti-tumor response, hopefully that can also prevent recurrence of really hard to treat cancers. So this is our image that was captured by our very talented technician, Ege Onal, who's now a MD PhD student at Brown University. And this is a glioblastoma cell. So glioblastoma is a brain cancer, and it has a terrible survival rate. I believe only 25% of patients survive past 1 year. So in this experiment, we wanted to see whether our mRNA nanotherapy was able to trigger the expression of an immunogenic cell death protein. So he captured this image 3 hours after treatment. And so here you can see this brain cancer cell that is in its death throes and undergoing this fiery immunogenic cell death. So as we're continuing to analyze the intricacies of what's going on and its implications for cancer biology, we're really excited about the intersection and the synergy between mRNA technology and our ability to harness this kind of immunogenic cell death, this powerful form of cell death to treat hard to treat cancers. So with that, I'd like to thank Professor Bob Langer and the entire Langer Lab, and specifically Ege and Jamie, who are awesome to work with. Of course, the Whitehead Microscopy Facility, where we took this image, and our funders. So thank you so much.
All right, we're doing it, guys. All right, thank you so much, Padmini. So as we continue for our story of the night, as we continue for the story of night, there is no greater challenge than the mind. And the journey through the mind's maze is the biggest challenge of all. To talk about what we find once we get there, Andrew Pickering will present his findings. Andrew?
Well, thank you for the introduction and good afternoon, everyone. I'm excited to share my image today and the work behind it, entitled Journey Through the Mind's Maze, Nanoparticles for Targeting Brain Cancer. Glioblastoma is a highly aggressive brain tumor that is notoriously difficult to treat. The first treatment strategy is usually to surgically remove the tumor. However, because glioblastomas are often highly diffuse and integrated into the healthy brain tissue, even if most of the tumor can be safely removed, there are often many tumor cells left behind that can then lead to recurrence. One treatment strategy that's being developed for treating glioblastomas, particularly those that are difficult to remove with surgery, is a direct local infusion of therapeutics into the brain. In this case, rather than going through the bloodstream, therapeutics must penetrate a dense network of pores between the cells in the brain, which in this image is represented as the red pathways. Each of these gaps is about 100 nanometers wide, which is about 100 times smaller than the smallest blood vessel in your brain. In the Hammond lab, we designed nanoparticle drug carriers that are designed to penetrate this system and specifically target drugs to cancer cells. We've engineered these nanoparticles to be about 100 nanometers in diameter so that they can freely flow through these gaps, but slowly enough that therapeutics are retained for long enough in the brain to have their effect. The objective of my research in the Hammond lab is to learn how different nanoparticle surface chemistries interact in the environment of the brain. In these 3 brain tissue sections, the yellow to red gradient is showing the distribution pattern of 3 different nanoparticles of similar diameter, but with different outer surface coatings. By designing the right surface, we can dramatically increase the distribution, enabling coverage of as many tumor cells as possible. Using a different technique known as flow cytometry, we can then analyze the cells in the brain and specifically isolate the cells that have taken up nanoparticles. And that is shown by the red dots in the inner box there. If we investigate this cell population further, we found that several nanoparticles enable increased uptake by tumor cells, which is shown in the red bar, and decreased uptake by healthy cells in the gray bar, which aims to improve the safety of this technique. If we zoom in at a much higher magnification, we can see this process of nanoparticles penetrating this dense cellular structures in the brain. The yellow to red gradient is showing the trajectory of nanoparticles as they travel away from the infusion site, between the cells with their nuclei shown in dark blue, and the cell bodies of the most abundant cell type in the brain, astrocytes, shown in cyan. This research was made possible through the support of my research advisor, Professor Paula Hammond, and the entire Hammond lab, specifically those who worked with me on glioblastoma projects. I'd also like to acknowledge the excellent core facilities here in the Koch Institute and thank our funding sources.
Okay.
I'm short, so I have to bring it down a bit. All right. Thank you so much, Andrew. This night is flying by like it was written in the astrocytes. Science joke. Which brings us to our next presenter, Michael Gallagher, who unfortunately cannot be here tonight, but he has a few words he wanted to share about his image. Across biology, structure and function are strongly related. The structure of the brain cells are especially breathtaking. Here, the astrocytes display the hallmark of star-like morphology for which they are named, with red and green labeling the expression of the 2 genes for their important function. To gain insight into the genetic risk for neurological diseases, the Yanish lab uses patient-derived cultures of microglia, sorry if I mispronounced that, neurons, astrocytes, which provide critical support for the neurons to perform large-scale, high-throughput screens to test the functions of different risk variants. I do want to make sure that the acknowledgement of the Whitehead Institute and the Yanish lab and the Young lab. So thank you so much for your contributions for tonight. Cancer is a communication between the immune system and the cancer cells. We are coming up on some magically communicating plants that will tell us about the dangers ahead. To speak more on this, our next presenter, Alice Boo.
Thank you, Kristen, for the introduction. So the image that I want to talk about, the story I want to talk about today is one about engineering the plant to microbe communication. In nature, plants and microbes naturally communicate by sending each other lots of chemicals. It's kind of like sending text messages, but with chemicals instead. And what we're trying to do here is to create a new communication signal between plant and bacteria that none of them can really sense otherwise. And we want to do that so that the microbes can help keep the plant healthy, but also detect things like landmines or heavy metals to help degrade them and restore contaminated landscapes. So how do we do this? We want to have a modular structure so that our microbes, our engineered microbes, can do different things and perform different tasks. So we want them to be able to sense different types of environmental stimuli, such as plastic. It could be nutrients in the soil, heavy metals, or pathogenic fungi, for example. And we use this by using a sensing computing output framework, which is kind of very widely used in synthetic biology. And so the bacteria will be sensing the stimuli, and then in response of it, will produce an output. And in this case, the output will be a bacteria to plant communication, so a molecule that will diffuse out of the bacteria and into an engineered plant cell. The reason we're using an engineered plant is so that no other plant will be able to detect the signal, only the plant that we're interested in. And so upon receiving the signal, the plant will produce an output. This output can be quite varied. It could change the plant phenotype, it could improve plant growth, boost the plant immunity, or degrade heavy metals, for example, that are in the soil. But for now, we're just focusing on changing the plant phenotype. We're looking at producing GFP, which comes from jellyfish and fluoresces and makes our plant green. So I'm going to focus on one specific example because we've been working with different plants and different types of bacteria. But this is an example where we're using a bacteria called Pseudomonas butida, which is a common soil bacterium, to detect arsenic, which is a heavy metal widely found in the environment that is toxic and causes quite concerns to human health and wildlife. So we've engineered our Pseudomonas butida to produce our bacteria to plant communication signal, here PCHSL, which we're producing through a 3-gene pathway. This molecule will diffuse out of P. butida and then be imported through just natural diffusion in Arabidopsis thaliana, which is a model organism used in plant biology. We know a lot about it. You can see it, a small version of it in well grown in hydroponic culture. And as you can see in the microscopy images, the bright field images shows the white structure of the roots. And when the bacteria and the plants are grown together with our arsenic, you can see that we're not detecting any green fluorescent signal in the root. But when the bacteria and the plant is grown together with arsenic, you can see a diffuse GFP signal along the root network. And you might say, well, this is great, like GFP is turning out in the root, but we don't actually normally see the root. When you look at a plant, you only see the arrow part of it. You only see the leaf and the flowers. What we're trying to do now is expand this work to be able to look at things in the leaves. So if you look on the left, the image is more in the violet blue tones, while on the right you have more orangey tones, which means that there's more GFP intensity that we're detecting in the leaf of those plants. So this still needs some optimization, and we're looking at using reporters that don't necessitate special equipment. So no fluorescent reporter, but pigments, which you would be able to just look at by looking at your plants. So with that, I wanted to acknowledge my PI and Dr. Tyler Toth, who worked on this project, and the whole Voight Lab for their incredible support. The imaging centers and the funders, and I wanted to finish on a cute note, which is not here. I had a cute doodle. Please, thank you.
All right, thank you, Alice. So every quest must face a challenge. Mucus rises up as an unlikely hero. It serves as the body's last line of defense. To tell you more about this sticky situation is Corey Stevens. Corey?
Right. Yeah, so thank you for the intro, and thank you to the organizers and the committee, and thank you all for being here. I'm excited to share my image with you, but I'm really more excited to convince you that mucus is interesting and fun to study. So most people maybe think of mucus as snot or something gross or some sort of waste product, but it's really, really important to actually, it covers all our epithelial cells that aren't covered by skin. So it's around 2,000 square feet. So it covers all our eyes, all our nose, all our lungs, our stomach, our GI tract, our reproductive tract. So around 2,000 square feet is covered by mucus. And if you pay rent here in Boston or Cambridge, that's really prime real estate. And it has a number of really important roles, right? It hydrates and lubricates these surfaces. So every time you blink or breathe or swallow, that's all facilitated by mucus. And it also acts as a barrier, right? So anything we ingest, any sort of virus or different type of foreign body, it has to go across the mucus barrier before it can harm our epithelial cells. And I'm interested in this barrier function of mucus. Mucus also has another sort of important function. It's a major home for most of our microbiome. So I'm sure most of you know we're colonized by trillions of bacteria, and most of those bacteria actually live within our mucus layer. So within our gut, many of microbes live there, and they do have important jobs like breaking down our food, producing vitamins, and protecting us from foreign pathogens. And mucus really carves out that environmental habitat that's home to most of our microbiome. So what is mucus actually? Mucus is really mainly composed of a protein. We've heard a lot about protein so far, but this is a sort of different one. It's called a mucin protein. It's a bottle brush-like polymer, we call it. It's bottle brush just because of the shape. It's what can be used to clean out glasses or vials in the lab. And we purify this from a variety of different sources. So from pig intestines or pig stomachs or human saliva. So if you're interested in donating saliva, please come talk to me afterwards. And what we do is after purification, when we rehydrate it and we look at that, this is what we see. And other than being this really striking image, what this is a rare glimpse of one of the major interfaces between us and our environment. This is the mucus barrier. This is what protects us from everything that we ingest. And in the best case, it prevents viruses from going across, but in disease cases like cystic fibrosis, mucus has gone wrong. So cystic fibrosis, irritable bowel syndrome, Crohn's disease, those are mucus-related diseases. But then other therapeutics we take, any oral therapeutic we take has to come across this mucus barrier. So how can we engineer things to get across this mucus barrier? And imaging this mucus barrier really helps us understand how it functions. We're also interested in how environmental contaminants, different types of pollutants that we ingest, how it damages this mucus barrier and leads to other sorts of diseases. There are mucosal associated cancers like lung cancer and ovarian cancer. That understanding how this barrier functions is really crucial to developing new types of therapeutics to treat those diseases. Also, mucus is just fun and cool. So with that, I was, great support from my lab, and this was done with 2 undergrads, and I think one might be here. I'm not sure, but Chris and Luke, and then the core facility here, this Peterson Nanotechnology, and David and Abigail work there, and lots of funding from different places.
Thank you, Corey, for that. All right. So now as the battle rages on, we have a hulking T cell who's going to come to the body's defense. To tell you about this unlikely defender, Keith Idell and Asla Gokdemir will tell you this tale.
So thank you all first to the organizers for this wonderful event. I hope everyone's having a great time. My name is Keith Idell. I'm a postdoc in the Human Laboratory right here in the Koch. And it's going to be a two-part talk with my able colleague, Asla. And the title of my talk today is Meeting at the Cellular Interface, CAR T cells versus B cell cancers. And, oh, we have it there. So I apologize. So patients that come into a clinic and present symptoms of leukemia, this is just a blood smear on your top left side here. And they will present symptoms of leukemia, which includes a number of B-cell malignancies that will override in the bone marrow and in the periphery of patients. The first line of treatment is often chemotherapy or cytotoxic drugs. Now, for some patients, they can achieve curative outcomes. But the very real and sad reality is that many patients will sadly undergo something called minimal residual disease, where disease burden goes down, but then over time they will relapse. These patients then become a candidate for a term that you may have heard in the news or here in Kendall Square called CAR T cells or cellular immunotherapy. And what this involves is that we can isolate out of a patient killer cytotoxic T cells. We can express and transduce in them particular chimeric receptors that will identify, target, and kill leukemic B-cell malignancies. Then after generating these cells, we can grow them up and reinfuse them back into a patient where they will travel throughout the patient's entire body, seeking, finding, identifying, and destroying leukemic cancer cells. And just as a schematic here on your left, so we have a T cell that's identifying a unique targeting cancer antigen such as CD19, and then it'll kill through releasing cytotoxic granules into the tumor cell. On your right here, this is a real-time movie of a green CAR T cell utilizing spinning disc confocal microscopy killing a leukemic cancer cell. In our laboratory, we particularly are studying BALL or B-cell acute lymphoblastic leukemia. And what you're seeing is actually apoptosis or cellular death or membrane blebbing. And one thing I do want to drive home here is that the actions are not from a distance, that the CAR T cell makes firm contact, and it's actually a battle between the receptors and ligands on one membrane versus the other. Now, when we stain for CD19 or a cancer targeting antigen, it's nicely on the surface. If we just take leukemic cancer cells and stain for a targeting antigen, it's nicely on the surface. However, when we add green CAR or engineered green CAR T cells and we mix them at different ratios, what we actually see in blue is our targeting antigen, is that these leukemic cancer cells have learned in a cell intrinsic mechanism of internalizing through a process called endocytosis, the targeting antigen. Additionally, in this image, depicted also with the yellow arrow, is that the green CAR T cells can physically remove the targeting antigen CD19, rendering the cell an escape mutant. And we also identify in these wells utilizing confocal microscopy that we get these big balls of green cells of CAR T cells that have trogocytosed or physically removed the antigen, and they are stuck together with like one red leukemic cancer cell. And we notice that the green CAR T cells are targeting themselves. So they are either exhausting themselves or killing themselves through a process called fratricide. And this will lead me to our awarded image that my able colleague Asla will describe what she did.
Good afternoon, everyone. I'm Aslı Gökdemir. I'm a visual designer. I'm embedding visual art into installations with data and science for public art. Thank you to Keith for sharing this meaningful data with me. I'm saying meaningful because it basically shows how human body fights with cancer. I redesigned the colors, tones, and textures to better convey the message of the impactful battle here. By distinctly separating cold and warm tones and focusing on the right hues, I aim to communicate the message effectively. I remodeled the image she took from the light microscope to show texture and depth by using Blender. Blender's features enable thorough detailing and realistic rendering, resulting in a visually compelling representation of the battle against cancer. And also, I aim to center the viewer's attention on the immune cell, vividly portrayed in bright green, symbolizing hope in this image. And circling it, warm tones depict the cancer cells, emphasizing the lumen tweet they pose. So that's how I put my perspective on this work. Thank you.
And I just wanted to add real quick for acknowledgements, all the members of the Heman Laboratory, including my three outstanding Europs, Joey, Hannah, and Anika. Thank you. On to the
Thank you so much, Keith and Asla. All right. So out of the darkness, a tower of hope emerges ahead, glowing promise of a brighter outcome. To talk about its promising hope, I invite Jolie Hung and Rhoda Sang.
Majority of recommended vaccines require multiple doses for full protection. As you can see in this table provided by the CDC, all of the recommended immunizations for children require 2, 3, even 5 dose administrations at specific time points. But requiring multiple doses poses challenges of missed or mistimed doses, and it can lead to serious diseases and even deaths. In fact, there are more than 1.5 million vaccine preventable deaths every year, and it's most prevalent in low middle income countries. As you can see in this map, there is an evident inequity around the world when it comes to infectious diseases. So in order to solve the global inequity caused by incomplete immunizations, we developed a controlled release injectable system that delivers multiple vaccine doses with a single injection. So here in this image, we have pink needle tip that contains first vaccine dose, gold core that contains a second vaccine dose, and blue base for the safety features. This is a completely dissolvable needle with a size appropriate for children that delivers 2 doses, eliminating the need for follow-up booster shots. This pink needle tip contains the first dose. This dried and solid needle delivers potent and effective vaccine without needing liquid injections. The needle tip dissolves in 5 to 10 minutes upon injection, and it initiates vaccine action immediately. This entire system is biocompatible and dissolvable, and yet robust enough for proper skin penetration, leading to no sharp waste. Okay. And the second part of the needle design is aiming to provide programmed future protection of the vaccines. So as you can see here in the center of this needle structure, the box is a microparticle containing 1 vaccine dosage inside. It is designed to give controlled degradation that can deliver vaccines over a monthly time window. And such control allows us to really achieve the tailored timelines that mimic the traditional vaccines. We also designed this slow dissolving base feature for the needle. So in order to ensure safe and secure vaccination, as you can see here, this blue part provides a base to the needle. It is made by polymers that can dissolve more slowly, and it allows the whole needle to maintain the structure integrity for a longer time after administration. So in the case of any allergic reactions coming from the first dose, we can retract the microparticle containing the second dose using the space feature. So in this project, even at the current academic research stage, we make sure to put the safety in the real world applications at the center of our design. And here we want to reiterate that in our lab, we develop technologies that aim to make vaccines accessible and safe for everyone in the world. Using one single injection platform, the vaccine protocols can be really simplified, and it can contribute to vaccine programs by lowering the healthcare costs and burden. And of course, we always aim to have enhanced safety with minimal and adverse impact. And finally, using the technologies, we want to develop innovation technologies that can bring equitable healthcare to the world. And finally, we want to take a moment to thank everyone, especially our advisors, Anna and Bob, and also all the fantastic members in the JACONET group. And we want to thank the Koch Institute, core facilities, and of course, our funding source, Bill and Melinda Gates Foundation. Thank you, everyone.
Thank you so much, Julie and Rhoda. Now we have Singyu Park talking about, oop. You didn't see this, this never happened. Right. Now we have Singyu Park talking about coming together in the end like a crystal mosaic to give rise to a better tomorrow. Singyu?
Thank you, Christian, and thank you all for being here tonight. My name is Sang-Yeon Park. I'm a PhD student in Professor Giovanni Traverse lab, who is also working in this building. And today I'd like to talk about one of my research projects on behalf of the team where we obtained this beautiful picture named Crystal Mosaic. So one of our lab's main research motivation is to support patients in achieving long-term drug adherence. As patients often fail to take medicine on time, still many medical problems stay unresolved, despite the remarkable advancement in pharmaceuticals. So one solution that gaining a lot of attention in this field is a long-acting injectable represented by in situ forming implant. This implant is initially a liquid form and can be injected through a needle in a minimally invasive way. However, once injected, they become a solid depot encapsulated drug in the polymeric matrix and achieve the long-term release of the drug. So this allows patients to achieve long-term therapeutic efficacy with just single injection. However, what we recognized was that this system mostly primarily relies on polymer to extend and control the release of the drug, which actually compromise the patient's acceptability. Let me elaborate on this in the following slides. So from this generalized plot, you can see that clearly the polymer loading increased the injection force exponentially compared to the same amount of drug loading. This means while the polymer plays a crucial role in the system for extending release, but at the same time, they inevitably limit the available needle size of the needle for injection, mostly larger than 21 gauge needle. And I put this figure to give a sense of this significance. So please consider the right screen as a reference and imagine how painful it would be if you try to inject something into your body through a 16 or 18 gauge needle. I'm pretty sure no one in this room would enjoy that experience. And besides, there has been many efforts to realize self-administrations for improved patients' convenience and acceptability. But such a big needle could be an even more significant problem for such self-adjustable applications, given the risk of traumatic experience and safety concerns on patients' end. So to overcome this intrinsic problem in injectable, our team has been developing an advanced long-acting injectable, where the primarily focusing on how to minimize the dependence on polymer. So in collaboration with Bill and Melinda Gates Foundation, we specifically aim to leverage this work to support people in sub-Saharan Africa to address their family planning issues by providing long-lasting, self-administrable contraceptive options. Now let's talk about the strategy. Our drug of interest, levonorgestrel, is a hydrophobic molecule, and the dissolution kinetics of its crystal form is primarily governed by surface area. So, so if you inject, without any polymer, and if you inject this crystal under your skin, then the duration of release will be comparatively short as that of a single crystal. So we figure, what if we are able to derive their compaction upon injection so that we can minimize their surface area and also extend the release. So based on that simple insight, we propose an injectable system where the suspended crystals are assembled upon injection and became a compacted crystal structure at the end, by which we can achieve extended release with a minimized amount of polymer or even without polymer. So from our benchtop study, we discovered that compacting assembly can be derived through solvent exchange. With a partially miscible solvent, the suspended drug crystals is dragged toward the environment radially, while they are entrapped by the partitioning interface, which is receding due to diffusion. And therefore, the drag drug crystals, along with the interface, is getting closer and closer over time. And at the end, they became a compacted drug crystal structure. And what matters is the solvent miscibility essentially determines whether the suspension can create a partitioning and receding interface with the environment. And depending on that, the suspension becomes just scattered particles or stay unchanged, like the top and bottom figure, or it can become a stiff solid implant at the end, like the middle figure, which can be even graspable with a tweezer. And if you magnify the graspable depot, you can clearly see the compacted structure which was assembled over the solvent exchange. So to make this interesting finding the translational, we conducted a preclinical study with rodent models. So here we injected the lead formulations as well as this negative control where the crystals are dispersed in saline. And as you can see clearly, our lead formulation showed 2.2-fold slow release, more extended release compared to the control group. And more exciting part is when we excise the depot and analyze the structure using micro CT, we found that our lead formulation showed significantly higher packing fraction compared to the control. So in this work, with our lead formulation, we achieve higher compaction, lower surface area, and therefore we achieve extended release as we hypothesized. And another beauty of this approach is we basically achieve this extension without any polymer, which also enable the injection of such a highly concentrated formulation through 28 gauge needle, which is 3.5-fold smaller than 18 gauge needle that I mentioned earlier as a typical size of the needle in the commercial products. So that's the condensed story and context about where this mosaic picture came from. This is another picture of the compacted drug crystals we obtained through solvent exchange. And based on the unique mechanism, we try to leverage this work to develop an advanced long-acting injectable. And from our animal study, we found that the more compacted structure from our lead formulation showed extended release in pharmacokinetics. And also, thanks to the polymer-free formulation, we were able to inject this condensed formulation through a smaller needle compared to the commercial products. So hopefully, at the end of the day, we are able to advance this work to develop a self-administrable, long-lasting implant for people in Sub-Saharan Africa to address their family planning issues. So with that, I'd like to thank my advisor, Dr. Giovanni Trevorso, and my mentor, Dr. Vivian Fai, for all the guidance and support. And this is today I had an honor to represent this work on behalf of the team. So I thank everyone for their own contributions. And also I thank the sponsor and the affiliated institute for all the fundings and resources. And I really wanted to shout out the amazing research engineer in the lab, Benjamin Mueller, who actually took that picture and submitted this work to the KI Image Award and make this happen today. So a major credit should go for Ben, and he should be also the hero tonight here. So thank everyone, and thank you for listening.
Mm-hmm.
Thank you so much, Seung and Ben. Went too far. So the beauty of a new day and new technology is where our adventure ends. Kusan Lu's image will lead us to a golden ending for tonight. Kusan, please come up and present.
Thank you, Kristen. Hello, everyone. My name is Kun Zan, and I'm a second-year PhD student at Professor Sixian Yu's group at the ECS. Optical microscopy is an indispensable tool as it provides cellular resolution that we can access the biosystems with subcellular information. However, imaging was mostly done in dead biosystems with limited imaging depth because of the need of the slicing and staining for providing contrast. In our lab, we are developing imaging systems that can illuminate living biosystems by engineering the light that can visualize the intact living system in real time with deep penetration and high resolution. The label-free nonlinear microscopy that we develop in our lab can visualize living systems with deep and dynamic functional and structural information based on their intrinsic molecular contrast instead of introducing labels. So for example, FAD and NADPH are two coenzymes that autofluoresce and are related to the metabolisms of the cells. We can see a clear distinction of the colors of the cells due to their different metabolic states in different types of the cells. Second, harmonic generation can reveal physical properties of a material, and this can be found in collagen fibers and muscle fibers in biological materials. While third, harmonic generation is sensitive to the changes of local refractive index, this can give us signals at the boundaries of the cells, including fat cells and many types of immune cells. While a single modality provides substantial information for the cellular environment, the integration of multiple contrasts or modalities creates a colorful depiction that reveals multicellular interactions. In this award-winning image, we multiplex these intrinsic contrasts that unravels the complex environment of the mouse whisker pad using intact, unprocessed tissues. Through the multiplexing of different contrasts, as well as their morphologies, we can differentiate different types of the cells, including stromal cells, muscle cells, fat cells, and immune cells, and identify the layered structure of the tissue, including root sheath, epithelium, basement membrane, and sinus. We can extract even more information of multicellular interaction by leveraging the three-dimensional imaging capability of nonlinear microscopy. For example, we can characterize the neuromuscular junction with precise three-dimensional localization of neural fibers and muscles. The integration of multiple contrasts provides opportunities to study the intricate multicellular dynamics inside its natural environment. And here is a collection of different organs and species we have imaged this year. The universal molecular contrasts allow us to depict diverse biosystems that can provide a fresh view with structural and metabolic information. As an engineering student, I will be working on further advancing the label-free nonlinear microscopy that would allow faster, deeper, and higher resolution visualizations of living biosystems. And with that, I would like to thank my advisor, Professor Sixian Yu, and our collaborators that push the limits of this microscopic technology together. Thanks for your time.
Thank you so much, Kusan, and thank you so much to our presenters. Can we give one more round of applause, please? And to the audience, if you've never played D&D, now you can tell people you have. So, a couple reminders, just quick. Don't forget to please, if you haven't already, take a snapshot of the QR code. You're voting for the People's Choice Awards, your top 2 favorites. That way you can, if you came here for your friend, you can vote for someone else's too. That way we make it fair. Also, as I mentioned, we are celebrating the 50th anniversary of the Koch Institute tonight. So part of that is building on a legacy. Our presenters tonight have been given an orange Lego, which in a moment, as we head to the reception area, I'll call them to come up and they will put the Legos down to set a foundation like they have already, and to put the Legos down to start us off. Then we will set the Legos of blue, teal, and white out for the audience. If you want to choose to sign the Lego to add your name to the legacy, you absolutely can. Again, we are here to celebrate the presenters tonight and their beautiful images. So feel free to add on and be part of this memory, not only for you, but also for them. And with that, I would like to again thank our sponsor, Fujifilm. I will announce at 7:45, well, maybe 7:30, since we're getting out a little early. At 7:30, who are the winners of the 2 Fujifilm camera kits. Please, quick reminder, if you haven't already, make sure you are signed in. If you need a wristband, please make sure to get one at the table. We will have the bar open. I guess we can open it a little earlier, and we will also have some food out and available. Oh, and this is news to the presenters. We have our tabletop set out in front, and there will be puzzles of the images that you see tonight. So feel free to choose a puzzle to do, get to meet some people, grab a drink, have some fun, and we will now release the doves or open the doors.